And welcome everyone. We're really delighted to be able to host this extremely important event. I think touching many areas of science. So this is on adjusting non-COVID-19 clinical trials in the era of the pandemic with a particular focus on statistical design and analysis. I'm Eric Kolachik. I'm director of the Boston University Hariri Institute for Computing. We're hosting this event. And with me are Joe Massaro, Ludovic Trincart, and Judith Lok. They are members of, and in this event is co-organized with, the Department of Biostatistics from the BU School of Public Health and the Department of Math and Statistics from the BU College of Arts and Sciences. So I'm going to give you a very brief intro because it's not me that you came to listen to. I'll set the stage a bit and then I'm going to turn it over to our speakers. Just a word about the Hariri Institute. So the BU Hariri Institute is focused on initiating research convergence and facilitating integrated initiatives, largely focused on social good at the nexus of where sciences and humanities meet computational and data science. We have strengths from everything from cloud computing to privacy and security to AI. And we are the strengths that we are as a function of the strengths across the University of BU, and very grateful for all the partners that work with us in this way. So about today's event, as of 1 year ago, I was curious. The source Statistica says there were over 300,000 clinical trials registered worldwide. As I'm sure any of you on the call must know from your own experiences, COVID-19 disruptions to daily life have translated in turn frequently to disruption of non-COVID clinical trials. And ultimately disruptions are going to mean statistical implications. So today's event was actually motivated from some of my discussions with faculty here at Boston University, where they asked, knowing I was a statistician by training in particular, do I need to adjust my statistical analysis for COVID-19 disruptions? And if so, how? Right? And being a statistician, but not a clinical trials person, I said, I don't know, but I know some very good people who do. So we reached out to these folks and they were very kind enough to agree to present today. We reached out to the departments and chairs of Biostat and Math and Stat. I'd like to particularly thank Jose Dupuy, the chair of Department of Biostatistics, for her help getting the message about this event out to the much broader community. So our speakers today are Professor Joe Massaro, Ludovic Trincart, and Judith Lok. I'm going to say just a word about each of them and then turn it over. So Joe Massaro is Professor of Biostatistics for BU School of Public Health. He has the secondary appointment in Math and Stat. He teaches the core applied clinical trial sequence in Biostat, for which he's won several teaching awards. He currently serves as a managing director of biostatistics and data management at the Harvard Clinical Research Institute, which is a contract research organization providing statistical and data management services for device, pharma, and biotech industries. And overall, he brings 15 years and more of experience in design and statistical analysis of clinical trials. Ludovic Trincart is an associate professor of biostat for the School of Public Health. His research expertise and professional interests focus on clinical trials, survival analysis, evidence synthesis, and meta-research. And he in particular brings knowledge generation in clinical research as an interest area. He's made particular sustained contributions to the methodology of meta-analysis of clinical trials and observational studies. And more generally, his work is in querying the process of knowledge generation through quantitative epistemology. And then lastly, Judith Lok is an associate professor of statistics in the Department of Math and Statistics in the BU College of Arts and Sciences. Her research program focuses primarily on causal inference methods and survival analysis, motivated in particular by HIV/AIDS-related problems. Particularly relevant to today's work is work she's done with colleagues on Learn As You Go, or LAGO, clinical trial designs, where the composition of the intervention package changes over time as earlier stage outcomes become available. And she's also done work on safety outcomes in HIV clinical trials that account for the presence of informative censoring, considering both overall effects and personalized medicine. So we've got a great group of speakers here to talk with you all. The plan is to move in 3 levels. We're going to start with Joe Massaro giving an overview of FDA and EMA level guidance, giving a sense as to what both the US and Europe has said in terms of guidance from the regulatory agencies. Ludovic is then going to talk to us about statistical issues and recommendations from more of a nuts and bolts perspective. And then the fact that we have to adapt here raises the interesting question as to whether formal adaptive designs and causal methods might be of utility here, and Judith is going to talk to us about that. They're each going to speak for 20 minutes. We're going to hold them to that. And then we're going to leave about 20 minutes at the end for Q&A. There is a Q&A function of the Zoom that's been enabled. So if you have questions that happen throughout, if you could just put those in there, we will be collecting those, and then we'll be either asking them directly or asking some function of them in the Q&A. So without any further ado, I'm going to stop sharing, and I'm going to turn it over to Joe Massaro.
Thank you, Eric, very much. I am going to share my screen to get to my slides. So I want to thank the Hariri Institute for asking me to be a part of this. I was very happy to accept, and I would like to thank everybody for taking the time to join. Just one quick clarification. I apologize, Eric. You've probably got a hold of an older update for me in terms of my background. I actually am no longer with the Harvard Clinical Research Institute. They're actually now called the BAME Institute. I wish it was 15 years of experience, but now it's been more like about 30 years of experience, so I'm a little older.
Oh, that was quite an old one, huh? My apologies.
But thank you very much. And again, thanks to everybody for joining. Very much appreciate it. Sorry. So first, I'm going to talk about the FDA and EMA guidance, just to give a little summary of what I feel are the most relevant points, as well as give a little discussion about my experience along the way for any points that came up. The FDA guidance is the statistical considerations for clinical trials during the COVID-19 public health emergence. It was written in March 2020 and updated in June 2020. And the link for it is given right here on this slide. Okay. So there's an introduction. The guidance, and anything that I have in quotes is really a direct quote from the guidance. The guidance outlines considerations for statistical analysis of primary and key secondary endpoints. That is the focus of the guidance in a trial affected by COVID-19 to help ensure the trial will provide interpretable findings with correct statistical qualification of uncertainty. As with any FDA guidance, as the guidances say themselves, they should be viewed only as recommendations describing the agency's current thinking. I think what's of paramount importance is that when protocol changes are going to be made due to COVID-19 or due to other reasons, that the FDA recommends that sponsors consult with the relevant division when considering these changes and changes to the statistical analysis plan that will impact the analysis interpretation of the results. Sorry. So one of the points on the guidance is trial integrity, okay? If you are going to make modifications to the ascertainment, to the collection of the primary and key secondary endpoint analysis, and we'll talk about how those ascertainment modifications might be made in a second. Well, it does require a protocol amendment or an investigational device exemption supplement. Modifications to the analysis of these endpoints will need to be made in the statistical plan prior to the database lock and any unblinded pre-planned interim analysis. One important point is any modifications that are going to be made to the analysis should not be based on looking at unblinded data that's been collected so far in the trial. In other words, not be based on comparison between treatment groups and see how that's looking, and then modify the trial according perhaps to results seen there. This increases bias in the final results and the type 1 error. Safety of participants is paramount, and in any changes to the ascertainment or analysis, and using an independent unblinded potentially data monitoring committee to assess unblinded safety on data collected so far and for the trial, remainder of the trial is acceptable and appropriate. Now, there is data that can be looked at, but it really can only be looked at ideally for all treatment groups pooled together as one group. And data that you can inspect according to the guidance in such a pooled, blinded manner are missing data rates and patterns, and especially missing data due to COVID-19. Very similarly, rates and reasons for treatment discontinuation or interruptions, and for premature withdrawal from the study. Site closures and what has caused the site closures, and disruption of treatment supplies. So these are just examples of data that can be looked at in a blinded manner in order to help try to assess the effect of how ascertainment of the data and analysis of the data should change. From an analysis perspective, I'm sorry, my cursor seems to have a mind of my up arrow, down arrow seem to have a mind of their own, and apologize for going back and forth between slides, but the slide I'm on now, in terms of an analysis strategy, stop the trial early is a possible strategy to consider before the original planned number of patients are enrolled, and conduct the final analysis. Okay. Now, one of the major concerns is loss of power. Okay. And because of that concern, perhaps you want to consider reassessing power in a blinded manner using the data collected so far in order to assess what the loss of power might be. And what I mean by reassessing power in a blinded manner, for example, for a continuous outcome, calculate the standard deviation of the outcome for both treatments combined. If that standard deviation is smaller than anticipated in the protocol, perhaps power is gained. And in a binary outcome, calculate the event rate for both treatments combined. If pooled event rate is larger than anticipated, perhaps power is gained. Now I'm speaking positively about what could gain power, of course, if the standard deviation is larger than anticipated, or the pooled event rate is smaller than anticipated, there could be loss of power. But it also depends on what assumptions you are making about the treatment effect. If for some reason you also want to change the assumptions without, of course, basing those changes on unblinded data, if you want to change your assumptions of effect size, and there is justification to do that, then that's something to consider as well. I have, in terms of my own experience, I have worked with a sponsor who has stopped a phase 2 drug study early due to COVID-19. And in a phase 2 study, the lack of statistical significance when you do the analysis is not as detrimental as in a phase 3 pivotal trial, where the results are used for approval. In phase 2, the results are used often to plan a future phase 3 pivotal trial. So it was felt and discussed with the FDA that the sponsor could stop the trial if they want. It was still expected that trends, because there was a large enough sample size, that trends, even if not significant at a two-sided 0.5 level, would be able to be observed with the lower sample size. I personally have not been involved in a phase 3 pivotal study that is considered taking this action to stop the trial early. But, prior to doing this, stopping the trial and doing analysis, you need to discuss this with the regulatory agencies involved in your trial. Because if you stop the trial and do the analysis without telling anyone, and the results end up being significant, then some people may get suspicious that you were looking at unblinded treatment comparisons periodically and decided to stop the trial when you saw a significant result. So it's important, if your plan of action is to stop the trial early and conduct the final analysis on the data you've collected, to obviously not look at the data before you make that decision in an unblinded manner, and to discuss with the regulatory agencies before that decision is finalized. Transparency is the key here, transparency of your decisions. Other things in the guidance: if a trial did not previously have an unblinded interim analysis to stop the trial early, perhaps you could consider adding one now due to COVID-19 and saying, all right, maybe results are looking very good and we can do an appropriate interim analysis, at this point, but of course with appropriate significance levels to control the type 1 error. Very similarly, if the trial did have a previous unblinded interim analysis before COVID-19 is causing delays, you could consider adding future or modifying a future interim analysis. But again, control of type 1 error is the key, and discussing of these plans of actions with the regulatory agencies is the key. One other thing the guidance says to consider is increasing enrollment after the impact of COVID-19 has passed, due to overcome loss of information. Now, that could be still months and months down the road. It all depends, on timing of vaccines and people taking vaccines and other things. So you have to kind of consider the timing when the impact of COVID-19 might pass, okay? But if you do this, increasing the enrollment to make up for maybe data that was not collected during COVID-19, you will need to run sensitivity analysis. And that's another common occurring theme. And this analysis later will be run when you do your final analysis that includes still the missing data with appropriate imputation methods. So if people are missing data due to COVID-19 and you enroll more patients to collect more data, well, there will have to be analyses done still, including everybody and including the patients with missing data with appropriate imputation methods. Now, that's not specifically mentioned in the guidance as far as I saw, but it's just based on my experience over the last 9 months or so. For binary outcomes, extend follow-up to include more events, because power for a binary endpoint really depends on the number of events that are experienced or the number of patients with events. Another type of sensitivity analysis you'll need to do on the last bullet point there is assess if baseline and post-baseline, even outcome differences exist between the patients you originally enrolled and the patients that you ended up enrolling post-COVID-19 to make up for the patients who were missing data during this COVID-19 pandemic. Make sure that the patients you enrolled all along the way are similar, is the bottom line. Missing endpoint data due to site closure during COVID-19. The missing data mechanism, if a site closes down, the missing data mechanism may not be related to any of the patient's underlying conditions. So you can consider removing all participants from your analysis. That should theoretically not increase bias. You should also consider removing patients, or can consider removing patients from analysis who didn't receive randomized treatment during site closure. These lack of data, lack of treatment are not due to the patient's underlying condition. The only problem is removal of patients, while may not increase bias in this situation, will have loss of power. And you may want to consider enrolling more patients after COVID-19 impact has passed. However, there is missing endpoint data not due to site closure. In other words, patients, for example, unwilling to go to site for a visit. And you can modify the collection or the ascertainment, remotely collect data if possible with online questionnaires or dictionary or interview-based clinician reports. Extend the ascertainment window. If the endpoint was to be ascertained 9 months after start of randomized treatment, plus or minus 2 weeks, consider extending the window for data collection to plus or minus 4 weeks. Okay. For composite endpoint, remove components difficult to ascertain and consider adding more clinically relevant endpoints that might be easier to ascertain. Okay. If you have a binary outcome that's based on a continuous or ordinal measure exceeding a threshold, consider changing the endpoint to the continuous or ordinal measure directly. This is another way to change your endpoint. That would add more power usually when you do that, when you change a binary outcome to continuous. Again, sensitivity analysis is the key. If you're collecting data in 2 different ways or several different ways, you have to make sure that the patients collected in the planned way versus patients collected patients whose data were collected due to COVID-19 issues are similar at baseline and post-baseline. And if they're not, then perhaps these remote or these modified, endpoint ascertainments methods may not be able to be used. The data collected may not be able to be used. Okay. The EMA guidance on points to consider for COVID-19 on methodological aspects of clinical trials is also from June 2020. Its link is giving there. Safety of patients is paramount. It's ethical to proceed. It's an ethical mandate to proceed with a trial that's been started so that the efforts taken by participants and physicians can benefit drug development and inform patient care. So it's an ethical issue in the EMA guidance that one should try to continue as much as possible to one should try as much as possible, to continue with the trial. It's an ethical responsibility. Again, if safety of patients is at risk, then that's an issue. Then, you may not want to continue. The impact on data collection, analysis, etc., is a case-by-case basis. There's not one global thing that is, approach that can be made in all situations in terms of modifying ascertainment or modifying analysis. Everything has to be done on an individual study basis. Sponsors should ensure that their existing systems are able to record protocol deviations and capture related reasons for those deviations. Data collection should not stop and should continue as long as possible, taking patients' risk, of course, into account. And again, consider alternative methods of data collection. So some of the FDA guidance and the EMA guidance share similar thoughts. Consider the risk assessment of COVID-19 affecting participants and the study, okay? And again, FDA and EMA guidance is similar in the fact that sponsors are advised to contemplate a blinded analysis on accumulating data to evaluate implications on recruitment, loss of study participants, ability to record data, interpret treatment effect, etc. Risk assessment should focus on reliability of the data from a trial perspective and should consider impact of intercurrent events. Use of unblinded data is not recommended. If you do have to do analysis of unblinded data, it should be done by conducted by an independent ZMC. And the purpose of such analysis ideally should be risk assessment and not to perform any unplanned interim analysis of efficacy. Okay. Again, but if you could control type 1 error, et cetera, and bias, then in general, as the FDA guidance discussed, you may want to consider adding an interim analysis. Okay. Change in the design of the conduct of trial should follow local regulations and be approved by an ethics committee. Okay. So the following slides are just my experience, but I believe I'm out of time, but they basically mimic what was ascertainment and collection methods and analysis approaches by the FDA and EMA guidance. And it just was examples of those the use of those.
Thank you, Joe. And I should mention for those listening, we will make the slides available and a recording of this available through the Hariri Institute website that we'll put together in the wake of this event. So moving on, Ludovic, it's all yours.
Well, thank you for organizing and thank you for inviting me. Are my slides showing correctly now?
Yes.
Thank you. So in this presentation, I'd like to discuss the consequences of the pandemic on randomized trials. And the pandemic has profound consequences on the conduct of clinical trials. First, as mentioned by Joe Massaro, the priority was to preserve the safety of participants and trial personnel. But there are also consequences on the integrity of trials with indirect consequences, for example, with patient enrollment being stopped or delayed, patients missing visits, or patients withdrawing from the study. There are also events that are directly related to COVID-19 when patients contract the disease. And so those events can induce modifications to concomitant treatments. There are also potentially effects on the rate of efficacy and safety endpoints. And the treatments for COVID-19 could also interact with the experimental treatment assessed in the study. So from the perspective of the clinical trial, it has consequences on data collection. I wanted to highlight this point first, with potentially the use of digital and remote methods to collect the data that were initially planned in the protocol, but also additional burden on data collection because we need to document those events that are related to COVID-19. And so it raises a number of challenges. For example, how to define accurately the beginning and the end of the pandemic period at a given site, or how to make the distinction between a confirmed case from a suspected case, and so on. So I'll assume that these COVID-19 related events are collected accurately. in that context, then the statistical question is, will our non-COVID-19 randomized trial provide a treatment effect that addresses the initial question from the protocol and the question pre-specified in the statistical analysis plan? Will the evidence be applicable in the post-pandemic world? So our community is still learning how to answer these questions, how to adjust analysis to the pandemic. But several groups have proposed to use the estimate framework, a novel framework, to assess these questions and eventually modify statistical analysis plans. So I'd like to introduce this framework in the next 3 slides, and then discuss at a very high level how to use it to eventually adjust analysis. So this estimate framework has been introduced recently. It's described in guidance by the FDA and the EMA, published in 2019. And the SD is to align the objective of the trial with the statistical analysis. So an estimate is defined by these 4 points: the target population, what are the treatments being compared, what's the endpoint, and what's the measure of treatment effect. And all that defines the treatment effect of interest in the study population for a relevant primary endpoint. So for example, in a heart failure trial, we focus on patients with symptomatic heart failure. We're comparing, for example, a sinus node inhibitor to placebo. The outcome could be the time to the first of cardiovascular death or heart failure hospitalization. And we could measure the effect using the hazard ratio. So really nothing new so far. There's a 5th component in this estimate framework. To define completely the estimate, we want to specify how we are going to handle post-randomization events. So what are the methods to define and deal with those intercurrent events? And this is where we can use the framework to think about our COVID-19 related events. So I'd like to make 2 additional distinctions. First, we're used to the intention to treat principle. And so what's the difference between estimates and this principle? It will depend upon the definition we retain for the ITT analysis. The possible definition is to analyze each patient in its randomization group. And the second point is we want to analyze all randomized patients. And so if that's the definition of ITT, then we can have estimates that conform to this definition or not. Among estimates that are conform to the ITT principle as described, generally speaking, there are 2 main categories: estimates that are called de facto and estimates that are called de jure. And probably the most prominent estimate, or the one we are used to, is called the treatment policy estimate. It's the intent is to estimate the difference in all randomized patients. So that would be the effect of randomized strategies, regardless of intercurrent events. You can also define an attributable estimate. That would be the difference in randomized patients attributable to the randomized treatment. So with the idea also that in de jure estimates that we can focus on the difference if all patients adhere. So the third point I wanted to make to introduce this framework, it's the distinction between patients withdrawing from the trial as compared to patients withdrawing from treatment. In the first case, it will lead to missing outcome data. And in the second case, treatment withdrawal, it will lead, it will not lead necessarily to missing outcome data. So that corresponds to discontinuing or deviating from the treatment as described in the protocol. But that doesn't lead to missing outcome data necessarily, because the recommendation is to avoid missing outcome data as much as possible. So the intent is to keep collecting data, particularly the outcome, even if patients discontinue the treatment or deviate from the protocol. So I wanted to highlight this because then we can see that to handle treatment withdrawals, we can address it via the definition of our estimate, and to treat or to address treatment trial withdrawals, we're going to address it separately through the estimation methods. So with that in mind, now we can use this framework and we can ask what's the impact of the pandemic on estimates. So on this slide, I note that for the four first point, population, treatments, endpoint, treatment effect measure, it should remain unchanged. I wanted to say that when we conceived the protocol before the beginning of the pandemic, the intent, of course, was to generate, to produce a treatment effect in a world without COVID-19. So it's not written in the protocol, but that was the intent. And so it seems reasonable to keep that objective as much as possible. Of course, we can think of many ways regarding how the pandemic is modifying these components. For example, thinking about the population, we could imagine that during the pandemic, participants who are enrolled differ from participants who were enrolled prior to the beginning of the pandemic. Participants could be healthier, they could be enrolled in specific sites where the pandemic has not started or is not, the infection rate is not as high. The age distribution is probably different. And so it's probably good to pre-specify as much as possible a comparison of baseline characteristics in participants, probably according to time periods defined around the pandemic, to see if actually we see differences in participants. We could also, I think, mention how COVID-19 affected the population differently. COVID-19 has exacerbated social inequalities. And so it's likely also that it exacerbated disparities in enrollment that have been documented. And so it would be interesting to pre-specify also subgroup analyses according to social determinants of health or demographics to see if the treatment effect is the same or not in those subgroups. We can also think about the treatment effect. For example, with time to event data, the censoring pattern may be affected by COVID-19. And so we can question the validity of the initial analysis. For example, the validity of a log-rank test relies on the idea that the probability of event is similar for patients enrolled early and those enrolled late. So we could question that and then modify the measure to use estimators that do not rely on the censoring pattern. So the 4 points here should remain unchanged, the idea being to address the initial objective of the trial. The fifth element, it's how to handle intercurrent events. And so this is where the framework, I think, can offer guidance. So the first step is to document those intercurrent events related to COVID-19. I listed very likely the top 3, COVID-19 infection, death due to COVID-19, and then withdrawal from treatment, that is discontinuation or deviation from the intended treatment. The first point, COVID-19 infection, it will generally be independent of the randomized strategy. And the impact on the outcome will depend upon patient characteristics and also the time of onset of the infection as compared to the occurrence of the endpoint. So typically, if the primary endpoint has already occurred before the infection, then there's no adjustment needed or no impact. Another distinction to make when documenting treatment discontinuation or deviation, it's whether it's temporary or permanent. And also try to assess if treatment withdrawal was directly related to COVID-19 infection or more generally driven by the pandemic, reluctance to go to the site, lockdown, and so forth. There's also, for those events, considerations about blinding, whether the trial was blinded, in which case it's fair to say that those events are independent of the randomization strategy. While in open label trials, we may think that those discontinuation or deviation are more likely in the control group. So how to handle those intercurrent events? The estimate framework that was introduced prior to the pandemic offers 5 different strategies. And so, again, several groups have described how to use the different strategies to handle events related to the pandemic. So the first strategy, it's the treatment policy strategy. It's the estimate I introduced earlier. And so the idea is to achieve the estimation of a treatment effect regardless of intercurrent events, right? So the post-randomization events are considered irrelevant to estimate the treatment effect. So this method is to be avoided for dealing with COVID-19 related events, clearly because the inference will not be applicable in the world post-pandemic. There's a nuance to be made, though, if the percentage of participants with post-randomization events due to COVID-19 is small, or if post-randomization events result in discontinuation of treatment are short, meaning a short period for treatment discontinuation. In those situations, this treatment policy strategy may be acceptable. I listed together these 3 strategies because they are not generally recommended. The first one is the principal stratification strategy. The idea is to identify subpopulations, and so it doesn't really handle or address post-randomization events related to COVID-19. The composite strategy, the idea is to incorporate an intercurrent event into the primary or secondary endpoint. So it might be relevant outside of the context of COVID-19, but clearly here, unless the trial is, for example, specifically about the use of ventilation, it would be inadequate to incorporate those events into the endpoint. The next strategy is called while on treatment strategy. It's particularly relevant for repeatedly measured endpoints. And the idea then is to consider the last measure of the endpoint before the intercurrent event. So again, it might work in situation where the treatment duration does not affect the treatment effect estimate. But in all, it's unlikely that these strategies help in the case of COVID-19. The strategy that has been recommended in the estimate framework is the hypothetical strategy. It consists in estimating the treatment effect, assuming that post-randomization event did not occur. That is, patients were able to complete the treatment, even in the case of an intercurrent event related to COVID-19. So these methods, under this assumption, work based on prediction, we are developing a model to predict what would be the outcome if the intercurrent event did not occur. So formally, it can be completely defined in the framework of counterfactuals or potential outcome. And it's probably a good strategy for most events related to COVID-19. Other strategies have been introduced, for example, by assuming that patients who experience an intercurrent event have the same outcome as if they had taken the control treatment. The general idea, again, is to come up with an assumption, develop a model, and then predict the outcome under this assumption. So now I'll address the second part. We had how to handle intercurrent event, and that's done by altering the definition of the estimate. How to handle missing outcome data, it's done through a statistical method. So different reasons why outcome data initially planned to be collected will be missing. Particularly, these two here will be considered independent, and we can have events related to COVID-19. So we are back to general consideration about missing outcome data, particularly the mechanism underlying the missingness, and whether the outcome is missing at random, including missing completely at random, or if this assumption is not tenable, missing not at random. And so to handle missing outcome data due to COVID-19, we can use conventional method to address missing outcome data. If the missing at random assumption is plausible, we can use direct likelihood estimation or rely on multiple imputation. If the assumption is not plausible, we can implement multiple imputation with, for example, a pattern mixture model. So I'll give two examples, a situation where missing at random assumption might be appropriate. We think about a cardiovascular trial. The endpoint is the time to a major adverse cardiovascular event. I'm focusing on the hypothetical strategy, meaning I want to estimate the treatment effect, assuming no treatment discontinuation. If the missingness is because participants didn't come to the site, they're concerned about contracting COVID-19, so they discontinue the treatment and ultimately withdraw from the study. So in that case, the participant withdrawal is independent of the treatment or the outcome, so the assumption is fair, and we can use multiple imputation. If these motivating factors are related to age, or other factors, we can incorporate these factors into the multiple imputation model. Now, another example, still in the same context, would be to think about participants who withdrew from the study because they contracted COVID-19, and thinking that they were treated and survived, then it would be not tenable to assume similar rates of cardiovascular events between participants with and without observed outcome. So in that case, we would implement multiple imputation by making an assumption about the missing not at random mechanism by specifying a higher rate of MACE in case of unobserved outcome. So I'll conclude with those messages. The estimate framework might help assessing the impact of the pandemic on the analysis. It's essential in that framework to document intercurrent events related to COVID-19. Handling these events is done by modifying the definition of the estimate, while handling missing outcome data is done by a statistical method. And most missing outcome data related to COVID-19 are likely missing at random. And I'll send you to this special issue of Statistics in Biopharmaceutical Research dedicated to COVID-19.
Wonderful. Thank you very much, Ludovic. Our third and final speaker today is going to be Judith Lok, talking to us about adaptive methods. Keep in mind we have a Q&A at the end, so feel free to begin putting things into the Q&A if you have questions. And again, if you came late, we will be making slides available. Judith, all yours.
Thank you, Eric. Let me share my screen. Can you see my screen?
Yes.
Okay. So thank you, Eric, for inviting me to give this presentation. I really appreciate the nice introduction, and you got mine exactly right. I have not been at BU for that long, so it's very recent. Thank you so much. And I am going to talk about adaptive designs and causal methods, and I will present to you some solutions that are already there in the literature, not developed for COVID-19, but hopefully useful in the era of COVID-19. I am Judith Lok. I'm an associate professor in the Department of Mathematics and Statistics. And here you have my email address in case you have questions about the things that I'm presenting here. As already introduced by both Joe and Ludovic, it's important to consider whether we are doing blinded or unblinded changes to the trial design. If the trial data was not looked at all, and one could in principle simply redesign the trial without looking at the data, but taking the COVID-19 situation into account. If there are any regulatory agencies involved, of course, this has to be done in consultation with those. What is more difficult is to change a trial after looking at the data, and especially after looking at the outcome data. Those situations are more complicated, although there are some solutions available. In general, whether or not you looked at the data beforehand, causal inference methods could help, in particular when data collection is interrupted by the COVID-19 situation. There are also available already some adaptive clinical trial results in the adaptive clinical trial literature, and I will go over some that may be particularly useful in this era of COVID-19. If for some reason your data analysis needs to be done more efficiently because due to COVID-19 there's less enrollment or there's more variation in the data, in that case efficient analysis of the data could help. And I will give you some literature on how to do that. What is the solution to the problem of analyzing or redesigning a trial impacted by COVID-19 depends on the setting that we are talking about. I took this picture from Kunz et al., who have been studying the COVID-19 affected clinical trials that are not addressing COVID-19 treatments, but other treatments, but that may, as both Joe and Ludovic indicated, be affected by COVID-19. Here we have the calendar time on the x-axis, and you see here the number of patients recruited on the y-axis. The line between here and here is the time that the patients stay in the trial. So this first picture here indicates a trial that has a relatively short follow-up, but that has almost enrolled fully at the time of the COVID-19 outbreak, but where some outcomes may be missing or impacted by COVID-19 when COVID-19 struck in March. The second picture is one where the follow-up is much longer. The enrollment period here is short. Everybody has already enrolled before COVID-19 struck, but the time that patients are in the study is much longer. And so still this trial may be heavily affected by COVID-19 because after COVID-19 struck, outcomes may be different. The last example is one where the time duration for each patient is relatively short, but where now instead of in this first time where almost everybody enrolled before COVID-19 struck, COVID-19 strikes somewhere in the middle. So you see that there is different situations, and from your trial experience you probably know, And it depends what solutions you can use, depends on what situation you find yourself in. So I will start with a situation where that is particularly relevant when the trial inclusion is almost done and the follow-up is short, and when the trial inclusion is almost done by the start of COVID-19. In that case, we could try to answer a causal question that Ludovic also already has included. What would have happened to the patients in the trial in the absence of COVID-19? And that's a causal question that I really like to answer with a causal inference hat on. In this case, one could censor the patients when they got COVID-19 or when COVID-19 interfered with their care. This doesn't mean that you don't observe the outcomes. It's that after COVID-19 strikes, you ignore their outcomes. And this is because after COVID-19 struck, maybe their outcomes would have been different. And if you want to know what would happen to the patients in the absence of COVID-19, we cannot use their outcomes directly. If we censor the patients when they got COVID-19, this leads to what is called artificial censoring. And this artificial censoring may depend on past observed patient characteristics. If it depends on past observed patient characteristics and not further on the patient's outcome had there not been COVID-19, this leads to what is called outcomes missing at random. And there are ready-made solutions for that in the causal inference literature. If artificial censoring leads to missing at random, so it depends only on observed patient characteristics, then inverse probability of censoring weighting can be used to answer this what if question. What if there had not been COVID-19? Inverse probability of censoring upweights those patients who are uncensored but were unlikely to be uncensored. And this is done so the weighted sample, sometimes also called the hypothetical superpopulation, resembles the original patient population. And there are results that indicate that IPCW leads to consistent asymptotically normal estimators for means, proportions, and in general, Z estimators. If artificial censoring, when patients get COVID-19 or their care was interfered by COVID-19, if this artificial censoring leads to a setting where there are no common predictors of censoring and the outcome of interest in the absence of COVID-19, then one can just restrict the analysis to those patients who have uncensored observed outcomes. This setting, where there are no common predictors of censoring in the outcome of interest, could happen in settings where the outcomes are censored for COVID-19 at a common calendar time. That is, if the patient's baseline characteristics don't vary with the enrollment time. In such a setting, IPCW does exactly this. It just restricts the analysis to those with uncensored observed outcomes. The next thing that might happen is if the COVID-19 struck just before the end of follow-up, but earlier, it is possible that the original sample size can't be achieved. It is also possible that people may want to increase the sample size if they feel that the variance is likely increased due to a mixture of pre- and post-COVID-19 outcomes. So here we have two settings. It can be the decision to change the sample size is made without knowledge of the outcomes collected so far. In such a setting, one could just replan the trial because what we know now is no different from what we know at the planning stage, except that now we know about COVID-19. But knowledge about COVID-19 is what is called an exogenous variable. It is not related to any trial outcomes had there not been COVID-19. In such settings, in collaboration with the regulatory agencies, one could just redo the sample size calculations. However, sometimes maybe people want to change the sample size based on knowledge of the outcomes collected so far, Of course, here, conversation with the regulatory agencies is particularly important, or sometimes maybe your trial is not affected by the regulatory agencies. There exist adaptive design methods for pre-planned sample size re-estimation. And some of these methods are based on what is called the conditional error function. How does this work? One could change the rejection cutoff of the test that people wanted to use so that the conditional error with a new sample size is the same as the conditional error with a sample size that was originally planned. And I say conditional error, the conditional error, I mean the error conditional on the data that have been collected so far. If this conditional error is the same with a new sample size because the cutoff of the test has been changed, as it was with the original setup, with the original cutoff of the test, then the overall type 1 error rate remains at 5% if the original error rate was also 5%. It is also possible that due to COVID-19, the intervention package, if we are talking about a multi-component intervention, or the dosage, if we are talking about a treatment with a dosage, is likely not as effective, and the team feels that the intervention package or the dosage needs to be changed. If this decision to change the intervention is made without knowledge of the outcomes collected so far, one could, in principle, replan the trial in collaboration with the regulatory agencies. Because again, COVID-19 is an exogenous variable and not related to any trial outcomes. One could redo the design. It is also possible that the decision to change the intervention package needs to be made with knowledge of the outcomes collected so far. For this setting, Daniel Neville, Donna Spiegelman, and I have developed what is called Learn as you go, or LAGO methods. And Eric has already referred to that when he introduced me. These are adaptive designs for settings such as this, and we have originally developed these methods for binary outcomes. We have simulations and initial results available for regression and other Z estimators. In LAGO designs, one could adapt the intervention package based on averages and consistent effect estimates. So important is we don't change the intervention package or the dosage based on individual people's outcomes. We change them based on averages and estimates. Adaptive learn as you go, or LAGO trials, are for such sections, especially for binary outcomes for now, because the other results we have not published. Typically, there will be at least 2 interventions, and so outcome modeling will often be needed for estimation. If the intervention package is adapted based on averages and consistent effect estimates, the resulting estimators are consistent and asymptotically normal. And not only that, simulations indicate that these asymptotic results lead to good approximations for common sample sizes. Important is that with a control group, the exact type 1 error is preserved for the null hypothesis that none of the interventions has an effect. It is also possible that we don't want to change the trial, but we just want to change the analysis. And maybe because of lower enrollments or because of increased variance, there could be a power loss. If there are pre-COVID and post-COVID data, the resulting mixture distribution of the outcome may lead to increased outcome variance. And Kunz et al. Have also reported this. And this could lead to power loss. Efficiency gains can be expected by using modern statistical methods to analyze clinical trials, and those have been published. In a trial with short follow-up, there are roughly 2 possibilities. If there is block randomization, This in practice leads to stratification by a pre or post-COVID variable. If there is no block randomization, this in practice leads to a baseline covariate pre versus post-COVID. For example, in clinical trials with simple randomization and the probability of treatment being 0.5, ANCOVA turns out to be consistent, and this comes from Young and Ciatis, and even its standard error, especially when you use the sandwich estimator, is consistent. And this result was proven by a group led by Michael Rosenblum, even when the linear model is misspecified. So ANCOVA is typically used assuming that a linear model for the treatment, how the effect depends on treatment and how it depends on baseline covariates is correctly specified. But it turns out that for a treatment effect, this ANCOVA method leads to consistent and even consistent standard errors, even when the linear model is misspecified. So that means that precision can often improve by using models without jeopardizing the type 1 error or the asymptotic coverage when those models are misspecified. And in the data examples of the Rosenblum group, this actually led to 4% to 32% variance reduction. And you kind of get it for free. Even if your models are misspecified, you still get consistent and asymptotically normal estimators, but your variance may be greatly reduced if those models are more or less correct. For many outcome types, the Rosenblum group has developed statistical methods and software to operationalize this. And they also have preliminary results for stratification and for inclusion of inverse probability of censoring weighting for trials with longer follow-up. There is also an important paper by Zhang, Ciatis, and Davidian from 2008 that works for simple randomization and for Z estimators. And they report in their example a relative efficiency of 1.33 to 1.51, so considerable gain in precision. And also this gain in precision can be attained without compromising asymptotic coverage. I will end with futility and efficacy stops. If a trial was severely affected by COVID-19, it might have stopped enrollment. And one might face the decision, and I also refer again to this paper about COVID-19 interference in existing clinical trials. Should we stop the trial? Should we continue after COVID-19? If the decision is made without knowledge of the outcomes collected so far, one could redesign the trial taking COVID-19 into account and in consultation with regulatory agencies if those are involved. If one decides based on the outcomes collected so far, there is one thing you can actually do for free. You can stop for futility for free. Because stopping for futility doesn't lead to an increased type 1 error, since when you stop for futility, there is no strong conclusion when the trial is stopped. One could also consider stopping for efficacy. In that case, as Ludovic also remarked, one could plan an interim analysis and consider existing theory on alpha spending functions. One should consider the design before carrying out such analysis. And if investigators are somehow aware of outcomes, one should consider redesign by an independent group of investigators not aware of the outcomes. Or maybe in the organization leading the effort, there is enough experience with interim analysis that they have some kind of uniform way to do that. And that should be adhered to, because there cannot be an indication that the decision for the analysis was based to increase the probability of a significant trial result. That's all I have. So some causal things, sample size re-estimation, and changing the intervention package, and efficiency using the same data, or futility and efficacy groups. I have at the end of my slide deck that I will not show you here in detail, but as you can see, I've collected some references for those who are interested in using these methods. You can also, oh, I shouldn't do that. That's not mine. You can also contact me if you like. And thanks again for coming. And thanks again for Eric and Catherine for organizing this important event.
Thank you, Judith. I'm going to go ahead now and move us to the Q&A period. I'm going to share the screen here just so that you have our speakers in your minds. Oops. And I think we have a handful of questions already. So some people are asking, can we share the slides, please? We will absolutely share the slides. We've also been recording, so we will share the recording. You can look to the Hariri Institute web pages for that in the next day or two. We have a question here from Lindsay Cohorn saying, I know that some clinical trials have switched to collecting their outcomes virtually. Does the virtual outcome need to be validated before that can happen? Any thoughts from our panelists?
I'm not sure what's meant virtually. If it means remotely, then usually no, in terms of starting to proceed with collecting the data remotely. Say it's like a questionnaire, quality of life questionnaire. That can be done remotely, and I've not had anyone been asked to validate the remote collection. However, they are going to have to do at the end, and maybe this is considered a type of validation, a sensitivity analysis to make sure that the data collected remotely is similar to the data collected not remotely.
Thank you, Joe. Any other comments, Ludovic, Judith?
I think there's sometimes just not much of a choice. If you cannot collect really, then you have to do what you can, because we have to make sure that the data are being used.
I agree with the previous comments. In some cases, the instrument used to measure the outcome remotely or digitally has already been validated. If you think about the measure of blood pressure curves that are distributed, we know are reliable. And I agree with Joe that a post-hoc analysis comparing the treatment effect for different types of measures is warranted. We could also imagine having delayed adjudication of events that were identified remotely.
Great, thank you all. We have a question here asking Judith, if you could explain more fully the ANCOVA approach that you mentioned briefly. Could you say a bit more about that?
That is just putting a linear model. So you're making it's especially useful for continuous outcomes. You model the expected outcome based on baseline covariates and treatment with a linear term for treatment at least. And you try to do that in such a way that you're trying to model the expected outcome given treatment and the covariates. But even when that model is misspecified, the resulting estimates that you get for the treatment effect, so for the coefficient that indicates the treatment effect, that is still going to be consistent. And the standard error, especially if you use the centers estimator, that standard error is also valid, at least asymptotically. So if you have normal data, these are exact results. If you have non-normal data and model misspecification, then these are asymptotic results.
Great, thank you. We have another question regarding causal inference, asking you to comment on the implementation, whether, for example, machine learning approaches might be utilized or Bayesian hybrid approaches. How much How much flexibility is there in what causal approaches one might bring to bear in this context that you can say something about?
So as far as I know, most of the asymptotic results rely on this model for censoring to be leading to unbiased estimating equations. So for example, this could be a logistic regression model. I think there are some initial results on Lasso, but I am not sure that is completely well developed. The important thing being that if there are no predictors that are joined for the outcome of interest and the censoring, then it should be well specified. So it should be flexible enough in terms of time if there is a time component. If there's no time component and it's just baseline covariates, then I have typically been working with fairly flexible logistic regression models, but there are other people who have looked at machine learning models for that. It is more and more common.
Thank you, Judith. We have a more general question here that perhaps begins to move beyond just statistics to also inclusive of communication in general, which is if you have a trial where recruitment is stopped due to COVID, and there's uncertainty, say, for example, regarding getting funds to continue at the latter date, and you have an investigator wanting to analyze primary outcomes now, how would you handle that conversation in that situation?
I think it needs to be kind of pre-decided whether you will later collect data again, so that you know what kind of level of kind of the alpha spending function, how much alpha can you use. If you plan to continue later, your alpha spending function should make sure that you don't use your entire alpha level, so your test cutoff will typically be more conservative than if you are not planning to do it. But at the same time, it depends also on whether this is all blinded or not blinded.
Yeah, and if the investigator truly wants to do the analysis now, the first thing to do is to reassess power in an unblinded manner at first. And if you reassess power in a blinded manner, as discussed in the Fda guidance, using some of the recommendations, then, and you feel comfortable with the power and stopping the study now, then I would do it. If you do feel, if it is felt that there is low power, then you either, you just have to be prepared. If you stop the study now, you might not get a significant result, and that it may be worthwhile to wait to see if the funding will be coming, if it can be assessed that the funding will be forthcoming at some point down the road. Otherwise, if you're uncomfortable with power, you're setting yourself up for a failed trial. Again, if the trial's exploratory in nature, maybe that's not as much of an issue. I don't know if this is an industry-sponsored trial or what, but if you stop a kind of confirmatory trial, then it risks the product being approved if you stop now and do not get statistical significance. So it's not an easy decision, unfortunately. And then if you do decide to do analysis now and have the option to continue, then I agree exactly with what Judith said to do.
Okay, I'm going to see how many of these we can still get to. We are finishing up at 2, which gives us about 5 minutes. Someone is asking about suggestions for analyzing data from unblinded pragmatic trials, comparing 2 standards of care strategies, but with different frequency, like less intense versus more intense, and asking, is it proper to adjust for the adherence outcome?
My answer to these types of questions is to frame your question really, really carefully before you start analyzing. And that is always the case when you're using causal inference methods. first phrase your questions really clearly. If you are interested in adjusting for adherence, your question may be a per protocol analysis, and you will maybe ask the question, what would have happened to all these patients had they fully adhered to the adherence protocol? And you will do that both in the treatment arm and in the control arm. And then you get most likely a different answer, but also a different answer to a different question than if you look at just compare those who were treated versus those who were not untreated, because that would be intention to treat. And typically intention to treat and per protocol answers are different because their questions are different.
Thank you, Judith. Any other comments on that? Okay, there's a question here, and it's asking if interventions change substantially when done in person versus remote. I think the in-person remote is a theme that's really impacting a lot of people here, so I'm trying to help us zero in on these a little. If the intervention changes substantially between those two, how can we account for this in the analysis if during COVID the intervention had to be done remotely for some patients? Is this more of a conceptual issue? Is there statistical aspects that one can bring to bear for an adjustment like that?
Well, I mean, I guess the issue is is this a randomized trial where both the control and experimental intervention are different if done remotely? Or is it a single arm trial where there's only one intervention? It's a very good question. I'm not yet involved with a trial that's had a different intervention remotely. It's either they do the intervention on site, or if they can't come on site, there's no intervention done. You know, the easy answer I'll give seems to be what I'm saying a lot, but you know, post hoc sensitivity analysis to just assess if the results are similar between the remote interventions and the on-site interventions. And if they're not, perhaps consider some way to adjust for it. I don't know if a simple model adjusting for it is appropriate. It very well might be.
Okay, thank you.
I think you could also maybe use the approach that I indicated is advocated by the group from Tiatis and Davidian, where they pose where you could make an outcome model that depends not only on treated versus untreated, but treated versus untreated before COVID versus untreated after COVID, and then see whether that additional term that indicates whether it's COVID versus non-COVID contributes a lot. And that could probably be also leading to consistent estimation if you use linear models using the approach by the group by Rosenblum.
Okay, let's see, one more question we'll try and get in here. I think this is aimed at some of what Ludovic said. The treatment policy strategy is considered the pure intention to treat approach, correct? Can you elaborate more as to why it is not recommended for COVID-related intercurrent events?
I agree that the treatment policy estimate is the one that's more in line with the traditional definition of intention to treat analysis. And in that case, the treatment effect estimate captures the intention, so the initiation of the randomized treatment, whatever happens beyond. For example, if you start chemotherapy and for some reason you cross to surgery. So this is what your treatment effect measures. And if COVID-19 events occur, this is what your treatment effect estimate will reflect. For example, if you have treatment discontinuation because of the disease, the treatment effect will incorporate that. And so this is not necessarily what we want to measure, thinking about application of the results beyond the pandemic.
Great, thank you all. And I see we're coming right up on 2 o'clock. So we're going to have to wrap up here, but I want to thank the over 150 people that joined us today. And I particularly want to thank our 3 speakers. Really an excellent set of talks. To let people know yet one more time, we will be posting the slides and making them available as well as the recording. We will do this with a follow-up communication that you should be receiving within the next day or 2. So thank you, Joe. Thank you, Ludovic, and Judith. Thank you very much. And goodbye to everyone.
Thank you, everyone.
Thank you, everyone.